Good evening and welcome to our sixth and final event in our 2024 Public Lecture Speaker Series. I'm Professor Susanne von Canberra and a Fellow of this Academy and one of the conveners of the series, along with Dr. Oliver Mayo and Associate Professor Raffaella de Michelis. Tonight we are on Ngunnawal country and I would like to acknowledge and pay respect to the traditional owners of this land as well as the lands on which our online audiences may be joining us from. As we share our own knowledge, teaching, learning and research practices, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of country. Thank you for being here tonight on the Shine Dome and thank you to those joining us online. Tonight we pay tribute to the work of Professor John Eccles. Sir John Eccles is internationally recognized for his remarkable, outstanding impact on neuroscience. He carried out his research in Oxford, Sydney, Dunedin, Canberra, Chicago, and Buffalo. His numerous scientific papers and books arising from detailed and pioneering experiment studies on synaptic mechanisms and the organization of neurons and mammalian systems continue to have a major influence on brain research. His writings on the mind-brain interaction generated a wide interest and debate. Eccles also made his mark as an administrator, particularly at the Australian National University and the Australian Academy of Science, of which he was a foundation fellow and the second president. Tonight, our two speakers will highlight the work of Eccles, his long-lasting impacts, and share with us how Eccles' legacy has inspired their own research. But before I go on, I would like to acknowledge some special guests we have with us tonight: Mary Menis, Sir John's daughter, and Jim and Monica, Sir John's grandchildren, as well as members of the Eccles family who join us online. Thank you all for being with us tonight and for continued support of the Academy. I would also like to make a special mention of Rose Manson, Sir John's eldest daughter. Rose was Australia's first female neuroscientist, and she worked closely with her father in the lab at the John Girton School of Medical Research. She co-authored 25 papers with her father, and a strong case can be made that Sir John's 1963 Nobel Prize was underpinned by his daughter's work. Rose passed away recently at the age of 95, and before her passing, she was honored by the ANU with an honorary doctorate of science for her significant contributions. Our condolences to Rose's family on the passing of a remarkable woman. Before I introduce our first speaker, a reminder that if you would like to join the conversation on social media, please do so by using the hashtag ScienceJourney. And as usual, we'll have a Q&A after both speakers at the end of the presentations. So if you have questions, you can ask those by raising your hand here in the theatre or by scanning the QR code that will appear on your screen during the event. So now let's hear from our first speaker, Professor Stephen Redman. Steve was a Professor of Neuroscience at the Australian National University until he retired in 2007. He started his research career in electrical engineering before switching to neuroscience and receiving his PhD in this field in 1967. He worked directly with Professor Eccles in Chicago in 1967. Please welcome Steve.
Thank you, Suzanne, and thanks to the Academy or those members of the Academy that were brave enough to ask me to give this lecture. And tonight I will start with a little story about Sir John Eccles and the work that he did to win the Nobel Prize. As Suzanne has told you, he had many appointments. Oh, I don't have to look up there, I can look here. He had many appointments. He was born in Melbourne in 1903. He grew up in country Victoria and went to high school in Warrnambool, and there's a science lab, or science block named after him there. He won a scholarship to Melbourne University where he did medicine. He won a Rhodes Scholarship to go to Oxford, where he worked with Sir Charles Sherrington on spinal reflexes. He came back to Australia in 1937 to the Kanematsu Institute at Sydney Hospital. Things were not so great there, and he went to be a professor of physiology at the medical school at Otago University in Dunedin. Now, the ANU was started in about 1950, and he was the foundation appointment in physiology as Professor of Physiology at ANU in 1952. And as he says in his memoirs, that was the beginning of 15 golden years, 15 years in which he made wonderful discoveries and had a most productive time and had a lab that was the envy of the world, and people, neuroscientists, came from all over the world to work with him. In 1967, first of all in 63, he won the Nobel Prize jointly. 1967, he went to the United States, first to Chicago, where I worked with him, then off to Buffalo, and then retired to Switzerland, where he died in 1997. The Nobel Prize was awarded jointly with Hodgkin and Huxley, and the citation is for their discoveries concerning the ionic mechanisms involved in excitation and inhibition in the peripheral and central portions of the nerve cell membrane. And Eccles' part of that prize was to work on excitation and inhibition in the central regions of the nerve cell, that is in the cell body rather than the axon. And I'll attempt to explain to you just what he did to win the Nobel Prize. But before I do that, I've got to give you a little bit of basic physiology. And the first is the stretch reflex, or the tendon tap reflex, which many of you will probably be aware of. That is, if you have your leg free to move and you tap the tendon, what happens is that there are receptors in the muscle called stretch receptors, and they're activated. They send nerve impulses into the spinal cord through this pathway and make synaptic connection with a motor neuron, and that motor neuron's axons go back to the muscle, and when the motor neuron is activated, impulses from that motor neuron cause that muscle to contract, and your foot kicks out in front of you. The same stretch receptor axons also make synaptic connection with an inhibitory interneuron, and when that interneuron is excited, it inhibits the motor neurons that innervate the opposing muscle, that is, in this case, the hamstring muscle, because if you want to contract the quadriceps, you don't want the hamstring muscle to oppose it. So that produces inhibition. Now, if you're going to study inhibition and excitation in the central nervous system, you need to know what's going on in those motor neurons. You need to examine the electrical signals that are happening. And these are pretty small items, and you can't see them. They're buried in the spinal cord. So the trick was to use a glass microelectrode to poke into them. This glass microelectrode is a thin capillary tube, hollow in the middle, and pulled out under heat until the tip reaches a very tiny point. It's about 1 micrometre in diameter at the tip, that's a thousandth of a millimetre, and it's hollow all the way down to the tip. And you fill it with difficulty with a concentrated salt solution. And that concentrated salt solution acts as an electrical conductor between where the tip is and where the recording apparatus is. So the usual salt solution was a potassium salt, because potassium has a high concentration inside the neurons. Then you don't have to pull on the muscle to excite it. You can, to excite the axon, you can stimulate it electrically, and when you do that, recording in the motor neuron to which it makes synaptic connection, first of all, there's a membrane potential where the cell is, the internal potential of the cell is 70 millivolts negative with respect to the outside. That's before any activation. And then when the nerve is stimulated, you see this potential here. If I can get this thing working for me. The potential on the right there is an upward going deflection, that is a few millivolts positive from minus 70 millivolts. It going positive in a nerve cell tends to move the potential towards a threshold, which will make this cell very excitable. And for that reason, Eccles called it an excitatory postsynaptic potential, or EPSP. If you then stimulate the nerve to the opposing muscle, that is the flexor, you activate the inhibitory interneuron in the spinal cord, and that makes this potential at the bottom. I can't get this out. Which is negative going. That tends to inhibit the neuron. It takes it away from its excitability state, and Eccles called that an IPSP, or an inhibitory postsynaptic potential. Now, in the normal activity of neurons, EPSPs and IPSPs are arriving all the time. They add up in very complex ways, and it's a subject of much investigation as to just how they add up, because each neuron does it in different ways. I'm going to attempt to explain to you the ionic basis for both of those synaptic potentials. But first of all, I want to show you what happens when you repetitively activate an excitatory nerve. The EPSPs add up, they sum, as shown here. And if you make the EPSPs big enough, they reach a threshold, called the threshold potential, and then a dramatic event occurs. The membrane potential suddenly goes positive. It moves by about 80 or 90 millivolts, and it lasts for about a millisecond, and that's the nerve impulse, or the action potential. And that action potential propagates down the nerve fibre from the neuron back to its target synapse, whereas the synaptic potentials don't propagate away from the neuron. And an inhibitory potential landed just about the time when an impulse was about to be generated, then that impulse would be blocked. Now, this picture is a very famous result. It's published in all the textbooks, and it's the result that won the Nobel Prize. And it shows the EPSP here, the membrane potential of minus 66 millivolts. And what's being done here is to change the membrane potential by passing current through the microelectrode. So the membrane potential is changed in a positive direction. When we get to about minus 40 millivolts, an action potential is generated. And if you keep going in a positive direction, that action potential mechanism is inactivated, and you can see that the EPSP is getting smaller and smaller, and at about 0 millivolts, it's not there. And if you go more positive than that, it reverses. So that event at about 0 millivolts is called the reversal potential for the EPSP. And that's the clue as to what the ions are that are generating it. And I've taken this simplified diagram from a textbook, and it shows the same result, that the EPSP goes to 0 millivolts. Now, inside a neuron, potassium concentration is high, and outside the neuron, the potassium concentration is low. So if there are holes for potassium to flow through, it will flow down its concentration gradient from inside to outside. For sodium, it's the reverse. Sodium has a high concentration outside the cell and a low concentration inside the cell. So if there are pores for sodium to flow into the cell, it will. It'll flow down its concentration gradient. So you might think, oh, well, it must be sodium ions that are doing it, because at the resting potential of minus 66 millivolts, the current is dominated by sodium ions moving into the cell. But that can't be the story, because if you go to 0 millivolts, here, there's still sodium ions flowing in, but there's no EPSP. So something's balancing the positive ions coming in. And that balance is done by the outward movement of potassium ions. So at the reversal potential, potassium ions are moving out, and an exact number of sodium ions are moving in. But the answer is, at normal resting potentials, the EPSP is dominated by the inward movement of sodium ions and a little bit of outward movement of potassium ions. Now, there's another ion that should be mentioned, and that's the chloride ion. Chloride is high outside neurons and low inside, but if you inject chloride ions into the neuron and do this experiment, it doesn't make any difference to the reversal potential. So chloride has nothing to do with the EPSP. But it does have a lot to do with the IPSP. The IPSP is generated by the movement of chloride ions from outside to inside, and no other ion is involved. Now, where do these ions move? When the synapse is activated, I'll show you the next little picture of a synapse. A synapse is formed at the swelling that is on the end of a nerve fibre, and it makes contact with the neuron membrane. And in this swelling, there are these little vesicles called synaptic vesicles, little spherical items, and they contain the neurotransmitter. And the gap between the synapse and the membrane of the neuron is very small. There's some ultrastructure that I can show you. And there is synaptic vesicles filled with the neurotransmitter, and in this case, for the excitatory synapse in the spinal cord, it's glutamate. And then there's a very narrow gap between the membrane of the synapse and the membrane of the neuron, about 20 to 40 nanometers. So on the picture on the right, you can see that when these vesicles fusing with the synapse membrane, that is, when the nerve impulse comes in, all kinds of complicated things that people study occur, and these vesicles fuse with the membrane and spill their contents into this tiny gap, and that's the neurotransmitter. It diffuses across the membrane and binds with receptors on the neuron membrane. And when that binding takes place, conformational changes occur in that protein and open pores for sodium and potassium ions to move. So that's the story of the EPSP and the IPSP, the ionic story. Now, I'll move on from Eccles and talk to you a little bit about my own work, but before I do that, I want to show you these famous drawings. These are drawings, anatomical drawings from a Spanish neuroanatomist called Ramon y Cajal, and he drew them in the late 1800s. And they're silver stains, and he'd sit at a microscope looking down at the tissue and draw them by hand. So first of all, there's the motor neuron with its cell body, and these branching structures growing out of it are called dendrites. They're hollow tubes. They contain cytoplasm, and they branch and get progressively thinner, and they get to be submicron diameters, but they spread out to about 1 millimetre away from the cell body. More dramatically is the Purkinje cell with a profuse branching structure. That's the principal cell of our cerebellum, and all that branching takes place in the plane of the slide. There's no branching coming towards you or away from you. And the third example I'm showing you is the pyramidal cell, which Lucy will probably tell you a little bit more about. That's the principal cell of our cortex, and again, it has quite a complicated branching structure. Now, when I was budding neuroscientist, the general feeling was, with the more established neuroscientists, that dendrites really didn't play much of a role in the activity of a neuron. And the reason that is, synapses out on dendrites would be ineffective in spreading their current down to the cell body where all the integration takes place. And the reason is that these membranes are leaky, so if you put current out on a distal dendrite, that current will start flowing towards the soma, but a lot of it will leak out through the membrane, just like a leaky water pipe would leak water if you put water in at one end and you wanted to get it out at the other end. Now, for the motor neuron, 90% of the surface area of the motor neuron is on dendrites, and 90% of the synapses formed on motor neurons are on dendrites. So it seemed to me crazy that we should ignore synapses on dendrites because of this so-called phenomenon. So with my colleague at the time, Bruce Wormsley, we set out to do some experiments to see what we could find out about the spread of synaptic currents in dendrites. And it shows here, the red is is an axon making synaptic contact with a motor neuron. Now, we recorded from that motor neuron and we stimulated that axon and only that axon, a single afferent fibre coming into the motor neuron. And we recorded the EPSP that it generated. And after we did that, we stained the motor neuron with a dye and we stained the axon with a dye and we reconstructed the connection. So, and the reconstruction is shown here. The axon is red, and the 3 arrows show where there are synaptic contacts. And you can see they're about 100 micrometers away from the cell body. And the EPSP it generates is about 200 microvolts in amplitude. I'll show you another one, the same type of experiment, and in this case the axon that we stimulated, shown in red, makes synaptic contact well out on the dendrites, about 500 micrometers out in the dendrites, and 4 synaptic contacts. And the EPSP we recorded in that motor neuron is shown below, and its amplitude is about 300 microvolts. In fact, it's bigger than the one that's generated near the soma. And also the other thing to observe is that the EPSP from the synapses here on the right, this EPSP here, is slower. Its rising time is slower and its decay time is slower. And the reason for that is that when the currents that spread from the synapses down to the soma travel through that dendritic tree, there's a lot of filtering going on. So the high frequencies are filtered out and the EPSP becomes slower. Now, it's possible to do this in reverse, that is to not reconstruct the connection, but to record the EPSP in a motor neuron and measure electrical properties of the motor neuron membrane and calculate backwards. Given how slow or fast the EPSP is, we can work out where its synaptic location is. The slower it is, the further out on the dendrites it's generated. And when you do that, you get this result. And there's a lot of scatter here, but what's shown is on this axis here is the amplitude of the EPSP at the soma, recorded at the soma. These are single axon evoked EPSPs. And on the bottom axis is the distance from the soma to the synapse in electrical terms, not in physical micrometers, but to give you an indication. About here is about 500 microns further out. So a lot of these synapses are well out on the dendrites. And the important thing to see from this result is that the amplitude of these synaptic responses at the soma does not diminish, does not go to zero as the distance from the soma increases. So this has been referred to by people who came after me as synaptic democracy. That is, synapses far out on the dendrites have as much say in depolarising the soma as synapses made on or near the soma. And the same result's been shown to apply for cells in the cortex and in the hippocampus. I'll show you one more result, and that is, again, work I did with Bruce Walmsley, and it goes back to that same picture I showed you earlier, where there were 3 synaptic contacts close to the cell body of a motor neuron. And we stimulated that axon many, many times, and those noisy records you see here are the responses that occur each time you stimulate. And they're noisy because they're very small. They're around about 100 microvolts. And there's instrumentation noise, there's noise in the neuron, and it's hard to see what the amplitudes are. And when you average it, you get this thing here. This is the average. And the record below that is the variance about the average, and there's a signal there. That tells you that the amplitude is varying about the mean. But we worked out ways of stripping that noise off and seeing what the actual amplitudes are. And here they are. This EPSP is bouncing between 3 different amplitudes, 100, 200, and 300 microvolts, with the probabilities shown. So to explain that result, one synapse has to be releasing all of the time because there are no failures. There are no examples where there are 0 millivolts. So one is releasing all of the time, 2 release, a second one and the third one release intermittently. You can use the probabilities there to work out what the probabilities of release are, and those probabilities are shown here. One synapse releases transmitter with all the time, the others release part of the time. And that's an important finding because it shows you how synapses can be strengthened. You can strengthen synapses by conditioning stimuli that will change the probabilities of release. If you increase the probabilities of release, then the synapse becomes stronger. Well, that's the end of my story, but I can't finish without going back to John Eccles and a couple of important people. And Suzanne's already mentioned Rose. Rose was one of the first PhD graduates from the ANU. She did most of the work for her PhD in Cambridge because when she came here there were no laboratories functioning. She had to go somewhere else, so she did the work in Cambridge. She came back and to work with her father, and she was the glue that made those laboratories work. She knew what the scientific questions were. She knew how the equipment worked. She knew all the dissections, and people were coming from all over the world. Some of them couldn't speak English. None of them had seen laboratories as sophisticated as these were, and she made it all happen. And belatedly, the ANU awarded her an honorary degree, as Suzanne has just told you, and that photo was taken on that day. And unfortunately, she didn't live much longer. But she was such a wonderful helper to her father. And the other person I must mention is a fellow called Jack Coombs, who was a physicist in Dunedin. He worked with Eccles in Dunedin. He came here and helped set up the laboratories, and he made these laboratories some of the best laboratories in the world in terms of stimulating equipment, recording equipment, stuff you just couldn't buy in those days. And finally, if you'll indulge me, I will show. That's what the laboratories looked like. They were not trivial to work in. And if you'll indulge me, this photo was taken in Eccles' office in Chicago in 1967. And that's his PA, Ellen, sitting there. And look, he was a wonderful friend to me at the time, and we had some lovely conversations, and I look back on those days with fond memories. Thank you very much.
Thank you, Steve, for your presentation and for sharing your work with us, and also the anecdotes at the end. We'll have the opportunity to ask Steve back to the stage later for questions, but first I would like to introduce our second speaker, Professor Lucy Palmer. Lucy is an ARC Future Fellow and an NHMRC investigator who heads the Neural Network Laboratory in Synaptic Biology theme at the Florey Institute of Neuroscience and Mental Health, University of Melbourne. Her research uses advanced techniques to investigate brain function, focusing on learning and memory formation in health and disease. Please join me in welcoming Lucy.
Okay, great. Well, I'd first like to thank the organisers for inviting me to give this talk. It's an absolute great honour to be speaking after Steve Redman, who gave a wonderful talk, and also to be speaking in honour of Sir John Eccles, who, you know, from ever since I first started neuroscience, I've always admired his work. And he's really been instrumental to the work that's done in my lab, but also the work that's done all around the world. Okay, so when I was first asked to give this talk, I was told that actually I am the scientific great-grandchild of Sir John Eccles, which makes me very proud. So as you heard, Steve worked with Eccles in Chicago, and then Steve had a PhD student, Greg Stewart, who was my PhD supervisor. And so I worked with Greg here in Canberra at the John Curtin School of Medical Research. And actually this is a photo that I was able to dig up from my PhD days, and you might be able to see that the microscopes that we use nowadays look very different to what Steve showed back then, a bit smaller. Okay, so every office of a neuroscientist has at least one book authored by Sir John Eccles. So as I was making this presentation, I literally looked up at my collection of textbooks, and there, sure enough, I have 3 of the books of Eccles, and I honestly do look at them a lot. And a lot of the works are still really relevant and really the foundational kind of basis to a lot of the work that we do in my lab. Okay, so just a little bit, before I get into the science, just to make sure that you're all on board with the synapse, and they're really the building blocks of the brain. So I feel like using this, but I won't. So you have the one neuron, and how it communicates with the other neuron is via the synapse. So you have a neuron, it sends its axon to another neuron, and it lands onto the spine. It's called the dendritic spine, and as Steve said, that's where all the action happens. So Steve also alluded to the very beginnings of discovering how neurons communicate with one another. And most neuroscience talks actually do have both a picture from Cahal and also an image from the recordings from Eccles. So this talk's much the same. So back in the 1880s, Santiago Ramon Cajal did some amazing pictures of using Golgi stains where he was able to visualise the morphology of neurons, and just by looking at their morphology was really able to deduce a lot about how neurons actually would function. So he called, you can see from this image here, that on these, what Steve described beautifully was these dendrites, which my lab looks at. Along these dendrites, there's all these little spiny protrusions, which are the spines. But Cajal called them nervous articulations. And it was when Charles Sherrington, a good, a few years, a decade or so later, coined the term the synapse. And then along the road to the discovery of how neurons communicate comes the all-important work of John Eccles, where, as Steve really nicely described, and I won't go into any detail, about how he was able to really understand the ionic flows underlying the potentials. So Eccles was really interested in, of course, how neurons communicate with each other and these EPSPs, or these synaptic potentials, and that work has continued up until today in my lab and in many labs all around the world, and we're finding out more and more about communication between neurons, and we've progressed somewhat, but there's still a lot to know. So what we do know now is that spines, or the synapse, is what we call plastic. That is, it changes, and it underlies a lot of how we learn and how we form memories. On your left, you can see that the morphology of the actual physical structure of a spine can change as it becomes activated. So if you go up to a spine, you stimulate it, then it actually grows in size. And this is only temporary. It lasts for, in this set of experiments, it lasts for a few minutes, and then it shrinks back down. We also know that synapses are also quite plastic in their function, and that is the size of the potential that Steve spoke about. So it all depends on the pattern of the input that comes in onto a given spine. They're able to change the size of the potential, and so essentially change the information that's been transferred from one cell to another. Okay. So a lot of the work that's done in my lab is that we're really trying to understand how synapses function in the living brain, in the intact brain. Most of what we do know is from cultured neurons or neurons that have been removed from the brain, and we're really looking at them in the whole brain when the entire brain network is still intact. So the human brain has billions of neurons and trillions of synapses. So you can imagine this is an immense computational machine that we have up here. And here's just images that only show neurons actually from different brain regions. And you can see it's absolutely jam-packed. So you've got the somas, and then you've got the dendrites and the axons and all the neuropil. Within this image, it's only neurons, and there's a lot of other different cell types. So it's really densely compact. And of course, my favourite structure, the dendrite, and Steve's favourite structure, is there amongst all this, and it has all the spines and all the synapses that occur. So something that we know and that I always find baffling, and I must admit this is always my favourite slide to show, is that single neurons, they actually receive thousands of synaptic inputs. So here are neurons from different brain regions, from different species, and you can see that their morphologies, it's all beautiful, but it's really defined by their dendritic arbor that receive all the synaptic input. And they don't just receive one or two synaptic input. So, for example, the pyramidal neuron, which is the work that I work on in my lab, that guy, number C, actually receives 17,000 synapses. So at any given time, they've been absolutely bombarded by information and connectivity from different neurons. So we know that the computational capabilities of the brain is immense, and even the computational capabilities of a single neuron themselves is also immense, just by the sheer numbers of inputs that they receive. Okay, so what do neurons do with this? And I won't go into detail because Steve described it really nicely, but it's the basis of a lot of work in my lab, is that they integrate this information. So if you are recording from a dendrite and you activate a bunch of synapses, then you get the EPSPs, or the synaptic potentials. Then at a certain threshold, if you get enough of the input coming in, then you get this event that's called a dendritic spike, but it's basically greater than the sum of itself. So dendrites themselves are able to receive information, they're able to transform this information and ensure that it creates a larger voltage event than the input that they actually received. And the importance of this is actually quite immense, because what it means that input that arrives from different locations along this dendritic tree, which in humans, our pyramidal neurons can be 1 millimetre in length, which is a long way for a potential to travel. But they're able to have an influence on the soma, and that's where action potentials are generated. And action potentials are the way that cells communicate through with one another. So by dendrites doing their thing and taking input and transforming it into this large event, then they're able to actually ensure that this information has been transferred to a neighbouring cell. Okay, so we're really interested to see how dendrites and how spines function in vivo. And so you might be asking, well, how do you record from such small structures in vivo? You can use the electrical recordings, but that doesn't really give you the specificity of a single cell. Probably a couple of decades ago, there's been huge advances in the use of imaging. So what we're able to do is that we're able to fill single cells with an indicator, a fluorescence indicator. Either it's a dye or it's actually genetically encoded indicators, which are used more these days. But in this case here, it's a calcium-sensitive one. So any time that the cell is active, you get an influx of calcium ions into the cell, and this indicator that we've plopped into the cell will fluoresce. So we're able to actually visualise whenever neurons have been active, whenever they've actually generated an action potential in response. So you can see flashing lights here, and they're the dendrites of a single cell in a mouse who's walking around and learning things, and I'll tell you about that in a second. So another huge advance is that coupling the advance in the indicators that we're able to use with the microscopes that we use, which are smaller nowadays. And I won't go into any detail, but we're able to image deep into the brain, or deepish into the brain, into the cortex at least, by using a technique known as 2-photon microscopy, where you essentially have a long wavelength laser light that's able to penetrate deepish into the brain, and so then you're able to really visualise. And so these kind of techniques have really kind of advanced our knowledge of neuroscience probably for the last decade or two. Okay, so with that, by using all these fancy, powerful techniques, we're able to ask a lot of questions, and there's been a lot of research and a lot of papers have come out consequently. So our knowledge of how the brain works is really expanding, but we still don't know a lot. And something that, for example, just to show, is the use of this technique to actually just try to understand neurons and how they act in the living brain, and when they're active and why they're active. So here's just an example showing the type of experiment where you're able to image from a dendrite in a living animal, and you present the animal with a stimulus. Here in the blue is what we call a heat map, and all the hot colours, so the yellows, means that there's been this calcium activity, and we're really able to understand what kind of information these neurons, or when they're active. And here, actually, it's a great example from what Steve was showing about the differences in the probability of release, is that we're able to see that neurons are typically not that reliable in generating action potentials in the living brain. They're what we call, especially in the cortex, sparse coding. So maybe 1 out of every 10 times they receive a stimulus, any given neuron will be active, and that might be why we have a lot of neurons, in order to ensure that we're fully kind of encoding our entire sensory world around us. Okay, but so we know that neurons are active in response to sensory stimulation. So we want to know where they're stored in the brain, or how they're stored in the brain. So how memories are formed, it's one of the great mysteries of neuroscience. There's been a lot of experiments done on it, and we know that memory is really important for making us who we are. And when our memory fades, then we do, have some terrible diseases like dementia, amnesia, Alzheimer's, and we all know what that is. It's a sense of people losing who they are. So memory formation is extremely important for who we are, and so understanding the neural basis of how memories are formed is really crucial to understanding the brain, and also what happens when it goes wrong. Okay, so you might know about the curious case of HM. We now know that HM is Henry Gustav Molaison, and he, in the 50s, he had his hippocampus surgically removed to control epileptic seizures. And this was I must admit, an unfortunate surgery that they performed on him. It cured him of epilepsy, but it actually took away all of his ability to create new memories. So HM, he could no longer form new memories, but he could still remember the prior 11 years of his life. So he was able to remember his childhood, but he wasn't able to form new memories. So we're able to look at cases like this and really get a grasp on how different brain regions, what role they're playing. So with this, we know that the region of the brain that they removed from HM, which is the hippocampus, which is now considered the memory centre, is really crucial in forming current memories, instant memories, like where you put the keys on any given day. But obviously memories aren't stored in the hippocampus. They must be stored somewhere else in the brain. So in my lab, we embarked on a couple of studies to look at where long-term memories are stored within the brain. So we know that the hippocampus connects quite intensively with a region of the brain called the cortex, and the cortex is usually associated with sensory stimulation and sensory encoding. But we started to think, and other people have postulated, that maybe the hippocampus is also a place where memories are stored. So they're made and formed in the hippocampus, and then after some time, a little bit hand-wavy, but in some time they then get shifted off into the cortex where they might be stored. So we embarked on a study to try to see if this was actually indeed true. So in order to do this, we wanted to actually be able to visualise the neurons that are actually forming memories. And we do this using a molecular labelling technique. I won't go into any detail, but it basically fluorescently labels neurons that have a change in their genome, which infers that they've just undergone a lot of activity. So what we did was that we taught a mouse just to associate a sensory stimuli with a water reward. And so it was very simple memory that they were learning. And then we went and had a look at the brain cells and whether any of them were active in the cortex or any of them were fluorescently labelled, inferring that they were active at the time of the memory being formed. And what we saw a lot of changes. I won't go into any detail, but we saw a lot of biophysical differences. But we did see that cells within the cortex were fluorescently labelled, so we know that they were involved in storing memory. And actually, interestingly, we know that they actually have more synapses because they opposed to cells around them, they had a lot more spines on any given dendritic branch. So we know that there are memories formed in the cortex and that these cells that they form the memories or store the memories have more synaptic connections that are involved. And then, of course, we wanted to see, we asked the question as to whether if we could just activate these neurons, are we able to activate the memory? Can the brain essentially remember what they've learned just by the activity of these neurons? So in this case, we turned to another modern technique called optogenetics, where you're able to essentially control neurons just with light. So it's a channel rhodopsin opsin that goes into neurons. You can shine blue light, and then these neurons will actually fire action potentials. So you activate neurons just by shining blue light on them. So we put this channel rhodopsin into those neurons that were storing the memory of this particular sensory association task, and then we simply just shine blue light onto the brain to see whether there was any we could retrieve the memory of that the animal had just learnt with the association. So this just shows that when you shine the blue light, you get action potentials. And what we saw actually is that we were able to, just by activating these neurons in the cortex, that we're able to actually activate and retrieve the memory that the animal had learned. So again, just more evidence that what we think the working hypothesis in this case is that memories are formed in the hippocampus, then they go up to the cortex to be stored and for later use for years to come. Okay, so just to summarise everything I've said here, is that the discoveries of Sir John Eccles are still contributing to modern day science, neuroscience today. And we now have advances in techniques that enable synaptic function to be further probed. For example, this image here is from an expanded tissue where we're able to really visualise spines. Spines are submicron in size, whereas we're able to now use different techniques. We were able to blow it up about 5 or 10 times. So we're able to get better grasp on the morphology, different to what Cahal did hundreds of years ago. We know that synapses are plastic and they change their properties according to different input patterns, and cortical dendrites encode perception, and they're also involved in memory storage. And optogenetics is a modern technique that opens new avenues of research into synapses and brain function. And so with that, I'd just like to thank of course, Sir John Eccles, who sparked research, my own research, but also the research of many people around the world. And then this is my lab in Melbourne, and this is our Christmas celebration, doing lawn bowls a couple of days ago in 35-degree heat, so it was lovely. So, and thank you all for your attention.
Thank you. Thank you. We'll stay here.
Oh, stay here.
We'll be answering questions.
Perfect.
Yeah. So thank you, Lucy. Fabulous talk. It was really interesting to see how new technologies really can make a difference to research. So you stay here, and I'd like to invite Steve to come back up so the audience can ask questions. Please raise your hand if you have a question for either Steve and Lucy, and someone with a microphone will come to you. And online audiences, if you can submit your questions via the form using the QR code. There's a question up there.
Hello, Stefan Breuer from ANU. I have a question to both of you. You have the typical conversation about a movie and then you try to recall the main actor and you can't get it out of your brain. And then someone gives a hint and all of a sudden you can remember the name of the main actor and the memory falls into place. So talking about Eccles and in his inhibitory neurons, is that sort of something inhibiting in your brain and then someone gives you a trigger and that opens some other neurons and all of a sudden the flow of information goes out and you can remember it? You know, is this how it works? What do you think?
Sounds like a good hypothesis. I mean, that's as good an explanation as I could give, but I mean, the answer is it's just unknown.
Yeah, I would probably to add to that, I'd almost say I don't think it's the inhibitory system that then releases the information. I think that there are pathways in the brain that's kind of activated and that you need to go through in order to get to a certain memory. And if you don't have that, you can't find that exact pathway, then sometimes it's a little bit difficult. But then by having a trigger, you're able to maybe find that pathway, and that the brain is very integrated in the sense that different senses all send, different parts of the brain all send, it's highly connected, so they all send information. So just say if somebody says, "Oh, remember the sound of this happened," maybe that pathway, which is a different one that you weren't tacking into in this first bit, became activated and then was able to kind of retrace that memory. But it's a lot of research going into that, and as Steve said, it's largely unknown how do we retrieve our memories, and also how memories are basically, you don't want to remember everything in your life. So yeah, how we kind of stop forming memories is just as important as how we form memories.
Is there an online question? No. Okay. Are there further questions people would like to ask? Are there? Just wait for the microphone to come to you, please.
How would you explain those people who have extraordinary autobiographical memories that do actually remember almost everything?
So a lot of, again, it's, yeah, I hate to do that, but how I would explain is that what I would almost say, it's not, they don't have, and I think studies have shown this, it's not that they have an amazing ability to form a memory, they just can't kind of dampen that memory or get rid of that memory. So they just remember it all. But we don't know much, if anything, about how we, get rid of memories, essentially. So I think that's the process, that people who are able to remember a lot, like that guy who flew over New York and was able to draw the entire, down to every building to precision. And that's something actually that the brain's obviously gone a little bit haywire, and it's probably not good for other aspects of life to be able to remember things in that much detail.
Here's another question down here. Oh. And green shirt. Thank you.
I was talking to my granddaughter a couple of years ago, and she was doing high school maths. And she said, "Grandma, what's the answer to this?" And I said, "Hang on a minute, it's been a while since I've done any of this. I need to get my cortex and my synapses operating again before I can answer." I was consciously thinking about it, how I could respond, which I did. So my question is, we're talking about sort of instant remembering of things that we'd in the past. I obviously had to think about remembering before I got to the answer. Can there anything you can comment about that?
Yeah.
You got it.
I was going to say it probably relates to that question too. It's trying to engage the parts of the brain that you need to in order to get to that particular memory, which we think someone's stored in the cortex, as we've got some evidence for it, but it's probably a mismatch all over. And there's something called the engram or the ensemble, which is it kind of takes into account the entire brain as being how a memory is formed. So we have a lot to figure out about that. Yeah.
Just thankful I remembered.
Yes.
There's a question online.
There's a question online from Dan W. and he's asked, what are recent advances in understanding dementia?
Dementia. That's actually a field that's had a lot of advances and a lot of research, but I'm probably the wrong person. I don't study dementia per se, just more so study the fundamentals. But what I do know is that it's actually that there's new drugs on the market, so they're really able to understand more of the neural mechanisms and maybe the ion channel changes and the changes that happen in the brain. And then excitingly, and which is a great advances, is that now there's new ways of treatment. Yeah, I don't think
so. My name's Jim. I'm John's grandson. And I just want to share one thing with you about an event that happened when Monica's brother Greg went over to see him in Contra. And in the course of the conversation, he apparently asked about me. And Greg had been down to our house in Birregurra, which is in Western Victoria, and he showed him a picture. And he said, "Oh, I've been in that house." And he was educated in Western Victoria. He went to a primary school just outside Warrnambool, but they used to have some yearly holidays to Lorne, and they'd come up on the train, stop in Birregurra, get on a jinker, get taken to the local house, which was our house, taken inside, given tea and scones while their luggage was put onto a carriage, and then the carriage would pick them up and take them on their journey to Lorne, which could take between 1 day and 3 days. But the interesting thing was that at the age that he was at, 95, 96, whatever, he remembered the house, and he said it was a colour photo, and he said the house was exactly the same colour as he remembered it. And I just thought that was just a nice thing to share with you all.
Yeah,
Thank you.
beautiful. I mean, memory formation, it's we really don't know how it's formed, and it used to be thought that it was at single inputs, EPSP, single spines, but now we're thinking that it's this ensemble and more the engram, in the sense that you require a lot of, almost a mini network. And those we do know that the cells that form memories are more tightly aligned with each other, and so they obviously can maybe last the test of time when they're, to ensure that memories are tightly in there. Yeah.
Hello, I'm Mary. I'm Sir John's daughter. I remember going to his study and he said, "Mary, if you've forgotten something, it means you've gone up the wrong nerve path. You've got to come back to the junction and go up the right nerve path, and you'll find the information." And I'm also interested in the word synapse, which my father said Sherrington made up, like Lucy said, but it came from the Greek, and I've often wondered what it meant in Greek. Anyway, does anybody know?
I'm pretty certain it has a Greek origin, but I don't remember what it means in Greek. You remember?
No, but I also, I did, yeah.
There's another online question, please.
We have a question online from Kirsten who's asked, we know the importance of potassium and sodium in the synaptic system, but what other chemicals are essential for this process? And what information can you share about brain food?
Brain food.
Brain food. Oh, goodness. I really can't answer the brain food, but I mean, these ion movements are just basic to how excitation and inhibition work. And I mean, I fudged the story a little bit because I didn't want to make it too complicated, but these ions, potassium high inside and sodium high outside, maintained, those concentrations are maintained by the expenditure of energy, because the natural process is for sodium to come in and potassium to leak out, and there've got to be pumps that pump them back again to maintain those differences in concentration. And those pumps require energy, and that energy comes from ATP, which is a product from breakdown of sugar. So brain food, sugar is an important brain food.
That's very good. Well, thank you all. This is unfortunately all the questions we have time for. A huge thanks to both Steve and Lucy for joining us tonight, and greatly appreciate you sharing your time and your knowledge with us. And thank you to my co-conveners of this series, Oliver and Rafaela. It's been a wonderful series to convene and to celebrate some of our earliest fellows of the Academy. And thank you all for joining us tonight, and all those who have joined us across 2024. We hope the series has left you feeling informed and inspired, and don't forget you can re-watch all the events via the Academy's website. And I'm now excited to announce the series for next year. It'll be AI in Science and the Promise, Perils and Paths Forward. As you can imagine, the series will explore the world of artificial intelligence, and we look at how AI is helping scientists make breakthroughs in health and medicine, climate research, agriculture, food production, and more. The series will also delve into potential risks, limitations, and ethical issues as AI becomes increasingly prominent in science and our society. The series passes are available now, so we hope that you will join us in 2025. Thank you very much for being here.